All right, so in the reteach, we talked about eukaryote versus prokaryote and how the root kary means nut, like a pecan or a walnut, and pro means before, and eu means beyond. So prokaryotes are before this kary appeared, and eukaryotes are beyond this appearing. So what it was is when they looked at through the microscope at cells for the first time, they noticed that some cells had this little thing in the cell and some did not. And that thing was the nucleus. So that's what they were calling the nut, like the walnut or whatever. Okay. Um, we talked about how all cells have DNA, cell membranes, ribosomes to make proteins, and cytoplasm. Okay, all cells have these things. We talked about how a eukaryote is defined by having a nucleus and membrane-bound organelles. Prokaryotes do not have a nucleus. They have circular DNA called plasmid that is just in the cytoplasm. It's not surrounded by a membrane. So some of the membrane-bound organelles that eukaryotes have are organelles, chloroplasts in plants, and photosynthetic bacteria. Well, maybe not bacteria, anyway. Chloroplast, mitochondria, Golgi apparatus, endoplasmic reticulum, either smooth or rough, vacuole, lysosomes break things down, mitochondria do cellular respiration and produce ATP. The nuclear envelope is a membrane that surrounds the DNA, so it makes up the nucleus. Eukaryotes do a process called mitosis to reproduce. Prokaryotes do something called binary fission. Basically, it's literally just copying the DNA and dividing the cell in half. The cytoskeleton is microtubules in the cytoplasm. It helps keep the shape, and in the case of like amoebas and stuff, it can help move the cell. It also helps move things inside the cell. All right, we talked about mitosis and meiosis. We didn't talk about meiosis a lot just yet. But mitosis, you start with diploid cells. They have 2 copies of each chromosome, and you end up with diploid cells. Meiosis, you end up with haploid cells, 4 haploid cells, one of each chromosome. All right. Mitosis is asexual. So this is how your skin cells, muscle cells, liver cells, eyeball cells, that's how they reproduce, is mitosis. Meiosis is going to produce your sex cells, or your also known as your gametes. So sperm, eggs, pollen, ovules, all that stuff. With mitosis, you're going to go, you start with a cell in interphase, and then you go into prophase, which the nuclear envelope starts to break down, and the chromatin, the uncoiled DNA, is going to start coiling and forming sister chromatids. The sister chromatids pair up and form chromosomes. During metaphase, the chromosomes line up and little spindle fibers attach to them. During anaphase, the sister chromatids are pulled apart. And then in telophase, you end up with 2 nuclei forming with the copied DNA, and then during cytokinesis, the cleavage furrow deepens and divides the 2 cells. The centromere is the part that holds the 2 sister chromatids together. These little things here are centrioles. They attach these spindle fibers and pull the chromatids apart. All right. Systems of movement in microorganisms. Cilia are these little hair-like things all around the cell, and as they wiggle, they move the cell. Kind of like you might move your arm through the water. That's how they move. Flagella is like a tail, and as it whips back and forth, or sometimes it kind of wiggles, it moves the cell forward. So that could be like a bacteria. And then pseudopod, pseudo means false, pod means foot, so it's false foot. This would be like amoebas. And so you might have an amoeba and it detects some food somehow. And then the next stage would be it extends its cytoplasm around the food. And then it has the food inside of a vacuole and it's going to digest it with the lysosomes. With a paramecium, you might have cilia for movement, an eye spot for detecting light. You might have a gullet for food going in. And this is called the contractile vacuole, and it's for moving water. All right. We talked a little bit about animal versus plant cells. Animal cells have centrioles during mitosis. Plant cells do not. Plant cells have a cell wall. Some bacteria have cell walls. Animal cells do not have cell walls. Both have a cell membrane. Both have a nucleus, mitochondria, all that stuff. Both can have vacuoles. Plant cells have one large vacuole called a central vacuole. It's important because it's that's when it runs out of water, it fills up with water and stuff, right? When it runs out of water, the cell wall doesn't have enough pressure on it to stay rigid, and the plant wilts. And so then you water your plant, and those central vacuoles start filling up again, and the plant can stand up. That's why sometimes you water a wilted plant, and it takes a while for it to come back because it's got millions of cells that it has to refill the central vacuoles. Plants have chloroplast, animals do not. We talked a little bit about acids and bases, talking about acid rain affecting a lake. If I have pure H2O, right, and I add acid, it's going to become more acidic, so the pH is going to go down. Remember, anything less than 7 is an acid. So these are acids, these are bases, this is neutral. 7 is neutral, okay? If I add acid to a system with a buffer in it, that buffer helps resist a change in pH, so the pH doesn't really change very much. If I add a base to a system with a buffer, again, the pH doesn't change very much. We talked about how photo means light, taxis means movement. So phototaxis is movement toward or away from light. We talked about the endosymbiotic theory, how there was a cell that was eating probably other cells, and then a cell that could make its own ATP from sugar, and one cell engulfed the other, but for some reason didn't digest it. And then because this little cell was making ATP, the bigger cell was able to survive more, and so it copied, they reproduced together, and that little cell became the first mitochondria. We talked about hypotonic, isotonic, and hypertonic. Hypotonic solutions have a lower concentration than the cell that you put in it. So that means there's going to be more water flowing into the cell than out of the cell, and the cell will swell and rupture. Isotonic, iso means equal. So, for example, if you have Gatorade and you put some cheek cells in there, they're isotonic to each other. So the water flowing into the cell and out of the cell will be the same, and so the cell just stays the same. Now, it may not live for very long, but concentration-wise, they're good. Hypertonic solution, hyper means above, so that means that the solution has more stuff than the cell. And that means more water is going to move out of the cell than into the cell, and the cell will shrivel and shrink and die. So if you take a fish from the ocean and put it in your freshwater tank at home, this happens. Okay? If you take your goldfish from home and you put it in the ocean, this happens. We talked about how diffusion was something going from high concentration to low concentration. Like someone sprays perfume on one side of the room, after a little while, people on the other side of the room will be able to smell it. We talked about osmosis being the diffusion of water. Passive transport just means that stuff can diffuse through the membrane, in and out, like water going through the membrane. Facilitated diffusion, there are proteins that help things move through the membrane, but there's no ATP needed. Active transport needs ATP. Okay, contractile vacuoles need ATP, so that would be active transport. We talked about how CFCs from old refrigerants, they actually banned Freon, styrofoam, and hairspray propellants, or any aerosol propellant, spray paint, or whatever. Now, CFCs and these things are banned, so they don't happen anymore, at least in the U.S. But the CFCs would get up into the stratosphere and they would interrupt the ozone cycle. And that's what caused the hole in the ozone. We talked about the cell cycle and how most of the cell's life is in interphase, where it grows, it replicates its DNA, it replicates organelles, and then it's ready for mitosis, prophase, metaphase, anaphase, telophase, and then it goes to cytokinesis and actually divides and becomes two cells. And we talked about how aerobic respiration uses oxygen, happens in the mitochondria. Anaerobic respiration does not need oxygen, happens in the cytoplasm. Fermentation, there are two kinds, lactic acid fermentation and alcohol fermentation. Lactic acid and alcohol do not need oxygen. There are different ways of splitting sugars to get some ATP. They're not as efficient as aerobic respiration. Lactic acid fermentation happens in your muscles when they can't get enough oxygen fast enough for the mitochondria to do this. The mitochondria keeps doing this, but it needs more ATP than it's getting, so lactic acid starts happening in the cytoplasm. Alcohol fermentation is what happens when you bake bread or you make beer or wine. The yeast gobble up the sugars, and as part of the process, they produce alcohol as a waste. And we talked about how if you get those problems where they're like, here's a DNA thing, give me the RNA thing, you got to make sure that you know what they're giving you and what they're asking for you. Because sometimes they might say, here's the DNA, give me the RNA. Other times they might say, hey, here's one strand of RNA, what's the DNA? Or they could even give you the DNA and you have to transcribe it to RNA and then translate it to protein. So you've got to make sure that you know what they're giving you. All right, I'll see you next time.